Okay, so we have the cell media pre-warmed. So it's in this water bath here at 37. The water bath itself is a little bit over temperature right now because I started it, but that's okay. I've got it with one of these little lead weights so that it doesn't tip over in there. So the first thing we're going to do, once that is warmed up, you could do it while it's warming up, but we're going to start up the hood. Just turn the light on, and I'll show this during the safety training as well, so it's not super critical to see everything. But you open up the sash. There are little marks that indicate where it has to go. If you don't put it at one of these marks, it will alarm. So then just take some ethanol, spray down everything in here. And then we can start loading the stuff that we're going to use for growing up the cells for next week. So some of the stuff, so we need these tissue culture flasks. So everything we put into the hood we need to spray down with ethanol first. I'm also putting in some 1.5 mil tubes. These are sterile. It's not critical for what we're going to do for them to be sterile, but those are going to be for just seeing how many cells we have using the hemocytometer later. And then we're going to need a bunch of pipettes. So you're going to put them here. So the 25 mil pipettes, we're going to do 15 mils of media in each of the flasks, so I have one for each of those. And then we're going to need to, there's about 1 mil of cells in the cell tube, so I'm going to have a 5 mil pipette for that. And then we're going to have to transfer volumes of cells out of there, so I'll have some more 5 mils for that. It's always a good idea to put a few extra so that you don't have to come out of the hood if you accidentally touch something and need to get a new one. You want to cut, and then I'm also going to put in some sterile 200 microliter pipette kits. That's for the cells, just testing the cells at the end. I also need at least one aspirator pipette for this, just to suck up any residual cells at the end. So these are the aspirator pipettes. They're basically like the serological pipettes, except they don't have any cotton in them because they're supposed to suck stuff up and into the vacuum flask. So I'll put 2. I should only need 1, but I'm going to put an extra one just in case. And then I will turn on the vacuum for the vacuum flask. So that is behind the biohazard bin. There's a vacuum for the building vacuum. Just turn that on. You want to be sure to check the level on the main flask. So this is a trap on the right side. The left is where the media actually goes. If it gets too high, it'll suck it into the trap. So you have to make sure that the media level in there is not too high. If it's too high, you have to bleach it and then empty it. I think we could cut. So you want to wait, around 10 minutes for the air to cycle a few times. You want to make sure that the flow indicator on the hood is green. That's how you know that it's actually established proper flow inside the hood, so that it's actually filtering. So I'm going to go into the hood to label the flask. So spray my gloves down with ethanol first. So if there's ethanol on the flasks, you won't be able to write on them. You might have to let it dry for a little bit. And we can cut. So once the flask, at least this part is dry, and you can wipe it off. But once it's dry, then we're going to label the flasks. We're going to put the cell type. We're doing 7F2 cells. We'll put the passage number, which we'll get off the tube once we take it out. I'm just going to label this high. I'm going to put the date and my initials. What's today? 6, right? So I'm labeling it on this part of the flask. This type has a little window here for you to write on. It may not be that way. But this way, whether the flask is in the incubator this way or it's in the incubator this way, you can see what this flask is. Okay, we'll cut. Okay, so once everything is ready to go in the hood, we're going to transfer our media. So take off the weight. This is going to be wet. That water is super gross. We want to make sure we get this nice and dry. And then we're going to spray it down with the ethanol and transfer it into the hood. Be careful because it'll be slippery. I'm also going to just put a tube in there for some tip waste. I'm going to put in there a 15 mil tube that I'm going to use for diluting the cells when we get to that part. Okay. So now I'm going to, before we get the cells out, I'm going to put the media into the flasks and into the tube that I'm going to use to dilute them. So, we'll get everything ready. I'm just going to loosen the caps. But I'm going to keep the caps on, right? So they're loose, but they're on so that even though we're working in the hood, everything is staying closed as much as possible. Open my media up, but keep the lid on there. get my pipette. So I'm going to open it. I'm going to open it, one, so that I can see the gradations when I put it onto the pipetter, but also so that I'm not actually touching the pipette. Right, I'm only touching the paper on the outside. So take it out carefully without touching anything. Take up 15 mils of the media. Transfer it to the flask. Then I'm going to put it back in the original paper just to keep any liquid contained. I'm going to put it in the waste for the regular trash. And then I'll do that for the rest of these. So now that I'm done, I'm going to close these, and that's just so that if I do accidentally knock them over, they won't spill, right? So when you're done with something, close it just so that you reduce the likelihood that when you're moving around in here, you might accidentally knock something over and it might spill. Now we're going to get out the cells from the liquid nitrogen. So they're in this. This is a doer. Basically like a very fancy vacuum flask that can store liquid nitrogen and keep things cold. So take out this styrofoam part. And then take out the cells. Now the cells that we want are at number 3 here. It can be difficult to get them out with the gloves on. So that I can get this back in there. There is a risk that it might explode, but for that reason, we're just going to take it into the hood and just open it for a second and then close it back up. And then we're going to thaw it quickly in the 37-degree water bath. So we just got to basically move it gently back and forth, watching for it to thaw. So this will take a little bit. It's done. It's completely liquid. There's no solid anymore. So we're going to wipe it off. Get the gross water off of it. And then we're going to go back into the hood. We have 4 mils of media here. Just getting stuff prepped so the caps are set on there. Use a 5 mil pipette. So I want to homogenize these by going up and down a few times. Make sure the cells are resuspended. and then I'm going to transfer them to the media. Go up and down a few times just to mix it up. And then, I guess I'll just leave it for now. So I'm going to open up my flask because now I can transfer it into the flask. This has to go in the biohazard waste because it touched the cells. Open up my flask, just set the caps on there. And I'm going to transfer different volumes to each one. There's about 1 million cells in there, so I'm going to transfer half of that, 500,000, into a high flask, half a mil into the medium flask, and then for 100,000, and then I'll just put the remainder into a low flask. There'll be a little bit left in the tip at the end. So that's about 3 mils, so I'm going to resuspend again, being careful not to suck it up into the pipetter itself. I'm going to put 2.5 in here. Put a 1/2 mil into this one. And I'll just put the remainder into there. Okay, so then I'm just going to rock the media, the cells in it, so that it can cover completely the bottom. I don't want there to be any empty spaces. You can tell which side of the flask is the bottom, but this is angled a little bit, it's angled upward. Only this bottom part is tissue culture treated that the cells can grow on it. Now that that's done, I'm going to transfer these flasks into the incubator. Alright, so this is set at 37 Celsius with 5% CO2. I'm just going to put them together in here, and then I'm going to let them grow. And I will change their media in a few days, because they need to have their media changed every 2 to 3 days for them to grow. So the last thing I'm going to do is I'm going to check to see how the cells actually looked. So I'm going to get a 1.5 mil tube. These are sterile, but they don't have to be for this because we're not going to actually use any of the cells in here. I'm going to use a 50 mil tube for the chip waste. going to take 50 microliters of cells here. Transfer them into the tube. Get rid of my tip in there. I like to use this because it contains the liquid, because that's biohazard to the cells. I guess I should label this tube so we know what it is. I'm going to put 7F2 on there. And then we've got trypan blue in the hood. So this is just for so we can see what cells are alive and what cells are dead. Going to add 50 microliters of that to the cells in the tube. Mix it a little bit. And then we're done in the hood. So I'm going to clean up before I take the cells out to look at them. So we've got a little bit of cells left. I'm going to aspirate that. I take out the aspirator pipette, and basically I just put the tubing onto the aspirator, take it out carefully, and then I just suck up the cells, so the vacuum is sucking them up. I like to put it up so that it gets everything out of there. And I'm going to put it back in the wrapper. Okay, that's biohazard, and I'm going to put this into this beaker that we're going to put bleach into, so we can clean out the tubing. I'll leave this tube in here because we can use that again. It's not full. And take out the biohazardous stuff, waste, put it into the biohazard. And then I've got some 10% bleach. I'm going to take that into the hood. I'm going to pour some into this beaker. Just enough, like, 50 mils, so that the tube can suck it up, clean out the tubing. And just leave the tube in there. All right, then I take everything else out. Spray down the hood with 70% ethanol. And then close the sash. So it'll alarm unless you get it to the right closing position, and then you can turn off the light. You can turn on the UV light and just leave that on for like 15 minutes to sterilize. And then anything that isn't biohazardous you can just throw in the regular trash. Okay, so for counting the cells, we are going to need to use this these microscopes. So 2 of the stations have this microscope, one has one that's a little bit different. So if students are working individually, they can just use the eyepieces, but there also are cameras that are attached to the microscope. So you have to turn the cameras on. When they're on, there'll be a little blue light, and you have to turn on the monitor, which is the bottom button in the back. You can also turn it around if you need to see, put the power buttons on the bottom. Oh yeah, and don't forget, there's basically a switch that changes the light between going to the eyepieces, going to the camera. You have to turn it to the camera for it to show up on the screen. It's only one or the other. You can't see both in the eyepieces and on the screen at the same time. So then, the next thing is we're going to look at the cells. I'm going to get out the hemocytometer. Right, so this hemocytometer has a special cover glass that will be with it. It goes on the top. Make sure you've got a waste for those tips because they are biohazardous tips. And mix it up a little bit. They're going to load about 20 microliters. It's not quite 20 microliters. Okay? And it'll wick in there. Don't force it. You're just going to use the capillary action to get it loaded. And then take out any excess so it doesn't spill. You can do both sides. You don't have to do both sides. We'll see how it looks. So you want to look at least to make sure that you're loading it onto the right spot. A little bit of like there's a little bit of dirt on here, maybe it was dirty when we put it on here. It's not ideal. But the live cells are these white things. So here you can see there's a few. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 here. Those are live cells. And then there's maybe 2 dead cells here. So the dead cells will appear blue because their membrane is not intact, and so the dye, the trypan blue, can get into the cells. And you use this for counting, basically. Right? So you just push down. And then we'll use that to calculate how many cells there were in the total volume. You usually just count So you're going to count the 4 corners, and this is in, there's a protocol that's in for week 2 for how to do this, but you basically count the 4 corners and then average that out and then multiply that. The last thing we need to do after we measure the cells is we need to clean the hemocytometer. So you've got cells on here, so it's biohazardous, so we're going to use 10% bleach first. And make sure that we get everything with that 10% bleach. And we're going to wash it off with just some deionized water. Then use isopropanol. That was the cover slip, and then we'll do the same thing. The hemocytometer itself. And then we're just going to set it on a Kim wipe and let it air dry. And then I'm going to change my gloves because that the trypan blue, even if all the cells are dead, the trypan blue is potentially carcinogenic, so I'm going to get rid of my gloves.